So in anatomy, you should have covered the cell. So we're not going to go into all the organelles and so forth because we're not going to be focused solely on all the organelles. But we are going to need to work with proteins. So we will look at certain organelles here. Mainly we're going to focus on protein synthesis and DNA replication. So we're using DNA. DNA is the code for proteins. These proteins are important molecules, so we'll focus on producing these proteins because we will need this. The other the other organelles that we will use will be mitochondria because we'll be doing cellular respiration, and through protein synthesis, you'll see we'll be using ribosomes, endoplasmic reticulum, and Golgi apparatus. But for the most part, a lot of the organelles, from a homeostatic, from a physiological standpoint, we're not going to be focused on a lot. We're going to be also spending a lot of time on the cellular membrane. So if you don't remember the cell, you might want to go back and just review it a bit because I'm not going to be going through a whole lecture over the cell. We're going to be focusing here mainly just on protein synthesis and DNA replication. So typically the DNA and the organelles involved in making the proteins. So first we'll focus here on protein synthesis, so making those protein, making those molecules, those chains of amino acids with that three-dimensional structure. And it's actually a two-step process. We have transcription and we have translation. Okay. Transcription's taking DNA and making RNA, and then the RNA translation is taking that RNA that is made through transcription, and chiefly messenger RNA, and using that messenger RNA to translate it into an amino acid sequence. Okay, so we got two-step process to get this protein synthesized. Because transcription, remember, our codes, our recipes for making our proteins are located in our DNA, in our genes, in our DNA. All right, so we don't use the actual DNA to produce a protein. We first make a messenger RNA. We just copy a small part of the DNA because the DNA is this very, very large molecule, and there are many genes. There are multiple genes on this DNA, so we only need to transcribe, make a messenger RNA of the certain gene for a certain protein. Okay, so these are the two steps. We'll go over the processes and the steps involved in doing both transcription and translation. All right, so our first step is transcription. All right, and to do this, we're basically taking a gene in the DNA. We're taking a gene in the DNA and turning it, copying part of that gene, or copying the gene from the DNA into messenger RNA. Okay. So where is this housed? We got to talk cell nucleus. Cell nucleus, remember, that's a ball inside the cell. Okay. It's got the little pores here. It is a double membrane, so it has two membrane layer. Okay. It houses this nucleolus, which we'll see is involved in ribosome production. But in here, this is where all those chromosomes are contained. Chromosomes with the DNA. The DNA has the code for the different proteins. Okay, a gene. So part of that DNA, the DNA, we have 23 chromosomes. Okay, the DNA, we have 23 pairs of chromosomes, and the DNA, each of those chromosomes has multiple genes on it. So we don't need to use the DNA to make a protein because each gene is a different protein. Okay? And we can see it can actually be multiple proteins, but we only need to use part of the DNA. So to make our protein, we just copy part of the DNA. We're going to turn it from DNA into messenger RNA. Okay? And you see the DNA is housed in here. We got a double layer. We'll see there are pores here, to allow that messenger RNA to go out and to do that second part, getting translation. And like I already said, DNA contains genes on it. We have 23 pairs. Humans have 23 pairs of chromosomes. And a gene is just a length of the DNA that contains a specific protein. Okay. And there are multiple genes on each chromosome. So there are thousands of genes, and we produce thousands and thousands of proteins. Okay. Then, like we said, the gene, the first step in making a protein is the DNA. That gene, that portion of the DNA, first gets transcribed into messenger RNA. If you remember, RNA is, what's the differences between RNA and DNA? RNA is single-stranded, where DNA is double-stranded. Okay. RNA uses uracil instead of thymine. And it has ribose instead of deoxyribose for the sugar, for that chain that is made. So those are the differences. Okay. We're going to take that messenger RNA to be translated. But first, let's look at this transcription. Okay, we're going to look at transcription, and we can see genetic transcription. First step, genetic translation, and overall, if you took both steps together, it would be called genetic expression. So when a gene is being expressed, the protein for that gene is being made. Okay? And this is kind of how cells operate. When they need a certain protein, the genes will be expressed. When we don't need the protein, typically the cell is not, this is a lot of work for the cell, so the gene will not be expressed. So the cells can change expression of proteins depending on what's going on outside the cell, what functionality, what they need to take place in the cell. Say if the cell is using a lot more energy, your mitochondria is involved in producing energy. So many of the genes to make proteins for mitochondria would start getting expressed. When the cell is not using lots of energy, we don't have to express those genes as much. So the cell can turn this on and off, this genetic expression. Besides housing the DNA and the genes, we saw that dark sphere in the nucleus. That is that ball within the whole nucleus. Here is a nucleoli. Okay. And we'll see this is involved in some of the ribosomal RNA. We'll be talking about ribosomes here because that is where that translation takes place. Okay. But the nuclei sits here, and the DNA is running out in the nucleus. So let's look at the actual packaging of the DNA. The DNA is this long molecule. All right, it was completely stretched out. It would cover a tennis court. So your cells are microscopic. So do you think this DNA can be stretched out into your cells? No. So we package this DNA into chromatin. Okay. We have proteins called histones that help package the DNA. And we'll see a slide of these, picture of chromatin in there and how DNA is wrapped around it. Okay. And the DNA and the associated histones with it are called nucleosomes. And there are two ways the chromosomes can be packaged in chromatin. If you remember, I said genetic expression. The DNA can be expressed, like the gene can be expressed, right, when we need it. Not all the DNA is being used by the cell at the same time. So when it's not being used, the DNA is going to be packaged. It's going to be wrapped up. Okay. When we need access to the genes, that's when the DNA unravels where that gene is, so we have access to the DNA. Okay, so when genes aren't being expressed, the DNA is wound up and packaged up. When the gene needs to be expressed, we can unwind that certain portion of the DNA where the gene is housed. Okay, when we are using the portion of the DNA that is being used, it would be in euchromatin. This is where we're actively transcribing. Okay, this is not wound up tightly. The DNA is not wound up tightly. In euchromatin, the DNA is unraveled, and now we can get in and we can make a copy of that gene on the DNA. When the DNA is not being used, when we're not transcribing, when we're not actively expressing a certain gene, the DNA can be wound up into heterochromatin. Okay, these are highly condensed. We have to have the DNA condensed so it doesn't take up the whole cell. All right, and so we have to wind this up. Okay, so two forms. Euchromatin is the actively expressed region of the DNA, the genes that are being expressed. Okay, that would be the DNA would be in euchromatin form. In the regions that are not being expressed, the genes are not being expressed, the DNA would be in heterochromatin form. So here kind of gives you the different portions, the different, not portions, but different variations of the DNA between euchromatin and heterochromatin. Okay. Here we have access. So the codes, we will see later, the codes are on the nitrogen bases. Remember the nitrogen base, if you go back to the chemistry lecture, these nitrogen bases, these are the codes for the different amino acids. All right, so if we want to express a gene, if we need to make a copy of this code, we need to have access to here so we can copy it. So here is a euchromatin form. The DNA is unwound. We have access to these bases so we can make a copy of them. Okay, they contain the codes or the recipe, if you want to think. Okay. But portions of the DNA that are not being used. Right? You can see first this DNA is wound up, gets wound around nucleosomes. Okay? And wound and wound and wound and wound. See here, we could have access, be similar to this, where we have access to the DNA. But as we wind, and we continually wind and we wind the histone proteins with those associated DNA, we can wind this very tightly and get this densely compact. This would be your heterochromatin. Okay, this densely packed. But when we need to have access to the genes, we want to unwind this, and now we have access to the DNA. We can make a copy of the genes here. Okay, so inactive form, active form. Okay. Euchromatin or heterochromatin. So you can see the different variations of the DNA. You can see where we can get transcription taking place in the heterochromatin form. Don't worry about so much on acetylation and deacetylation. Those are active, but I'm not going to hold you to that. What you do need to know is that when the DNA is active, right, it is in this euchromatin form. So we have access to the DNA to make copies, because we're going to take, our first thing is transcription. We're taking the gene from the DNA and copying it into messenger RNA. So now let's look at the steps involved in transcription. And transcription is going from taking the gene and the DNA and making a copy of that gene, just only that gene, into RNA, and specifically messenger RNA. Messenger RNA is the copy of the gene. So we're going from DNA to messenger RNA in transcription. We'll see on our DNA, we have a region called a promoter region, and this is where it signals a start of a gene. Okay, so the promoter region signals the start, and we're going to just basically copy the gene that is here in this DNA strand. Okay, the gene would be located down here. If you remember, the gene is the code for the protein, and the codes are in the various bases, the order of the bases. Remember, we had adenine, we have thymine, we have guanine, and we have cytosine, ATGC. Remember, the A always binds with the T on the other strand, and the G always binds with the C. The only difference when we're making the strand is if it's an A on the DNA, the messenger RNA is the complement. So instead of a T on the messenger RNA, you would have a U, because we use uracil instead of thymine. Okay, so the enzymes. We haven't talked enzymes yet, but enzymes catalyze reactions. And in this case, the enzyme is RNA polymerase II. RNA polymerase II. And what RNA polymerase do will actually unzip. It's this big, you don't need to know transcription factor 2E, transcription factor 2A. Just realize that RNA polymerase is an enzyme involved in copying the DNA into messenger RNA. That's the reaction that is catalyzed by this RNA polymerase. Okay. So the first thing the RNA polymerase, that enzyme, is going to do is, remember, DNA is double-stranded. So we can't make a copy of a strand unless we open up the DNA and to make it into two single strands, because one of those strands holds the gene for the protein. So RNA polymerase is going to break the hydrogen bonds between the bases. Remember, those bases, the two strands of DNA are held together by the hydrogen bonds between the bases from both strands. Remember, we have this, and we have a base here and a base here, and there's hydrogen bonding between those two bases. The RNA polymerase is going to break those bonds to open up. So then we can open up the DNA. Okay, because we have to get in here and make a copy of this region. If this is where the gene is. Okay, just one side would be the copy. Okay. After it breaks the bonds, it's going to add the nucleotides. So say this is the strand we're using. This is the strand that has the gene. We have A, T, G, C. The RNA that would be put in here, what would be the corresponding base? Instead of T, it would be U here. And with T, it would be A. Okay. Again, this is our RNA strand that we're producing. RNA. And G would be a C base, and then C would be G. Okay. In the various configurations along this gene, there's be hundreds to thousands of these base pairs that make up a gene. The configuration is going to be the code for which amino acids are going to be added. Okay, so that's essentially what we're copying, is the code for the amino acids. So as this gets formed, right, we get the bonds created by dehydration condensation reaction, and we form a single strand of messenger RNA that is basically a complement of whatever is in this DNA strand. Here is the RNA polymerase, this big circle. This is a large enzyme. If you remember, first it must unzip. Here is the DNA, we're unzipping. Okay, there is a template strand of DNA that has the code for the genes. Okay, that's what we're going to make the complement to this gene here. Okay, the coding strand. Okay, so the coding strand will actually be the same base pairs as the RNA we're creating. Because remember, here is the template strand. Okay, the complement strand is the coding strand of DNA. Okay. If this is A, then it would be T over here. So if it's A here, the corresponding messenger RNA would be U here. Okay, so this RNA strand basically is exact copy of this coding strand, except for T. Any T here would be made into a U in the RNA strand. Okay, so that's what the RNA polymerase does. It opens up the DNA and then it adds bases and catalyzes the bonds, that dehydration condensation reaction between the phosphate and the ribose. Okay, making the bonds here, and now we have the nitrogen bases. Okay, because we're going to see these nitrogen bases hold codes for certain amino acids. As we look into the further steps of making RNA, let's look at different type of RNAs we'll find in the body. Okay, there are more than this, but kind of this will keep us where we need to be. Is we've already seen messenger RNA. Messenger RNA is that code. That is the copy of the gene in the DNA. We will see later transfer RNA. Okay, transfer RNA, tRNA, is going to be involved in translation. We do add the amino acids to the messenger RNA strand when we translate the messenger RNA. And then the ribosomal RNA will be found in the ribosomes. Okay, that's the site in which we're going to be doing protein synthesis or translation. So the transcription, making that copy of messenger RNA from the DNA, takes place in the nucleus. Right? Then our messenger RNA has to go out to be translated. It goes out to either the endoplasmic reticulum or into the cell. But that initial RNA that is made, it is considered precursor messenger RNA. Okay, because it will be altered. It will be altered before we go into the second step and before we go into translation. Okay. Because in that gene, there are portions of the gene that do not actually code for proteins. They don't actually contain the codes for the different AAs, the different amino acids. Okay, so we're going to be doing what's called splicing out those portions. Right? And so the portions that are kept are exons, and there are multiple exons in a gene. And we can see we can splice out different exons, and we can actually make different proteins from a single messenger RNA, depending on how we splice it. Okay, so let's take a look at this in the next couple slides here. Okay, so that pre-mRNA, that initial mRNA, is not the mature RNA. It's not the RNA that's going to be translated. So first, we have a spliceosome that actually is going to splice out the introns. The introns are non-coding. The exons contain the codes for the amino acids. Okay, so we splice out the introns and put the exons together. So here's kind of the overall step. Okay. You can see this is our first step, our transcription. Okay. We're making this RNA. This is our template strand, and here's our coding strand of DNA, and we're making a strand of messenger RNA. Not the mature though, because remember we got to take this RNA, the initial RNA, and so here's the fully transcribed RNA, and now we're going to splice out those introns, those non-coding regions, and splice together the exons, okay, to make the mature RNA. This would be the RNA that would go and go to the ribosomes and do that second step, which we're going to go over, that translation step. Okay, so let's finish this transcription part, or getting our messenger RNA. So we can see there's genome and proteome. These are basically the gene. How many genes? What genes are there in the body? And what's the proteome? Right? Because we do say a gene is a code for a protein. But we will see there are much more proteins made in the body than there are actual genes. And how can this take place? Well, when we make our initial, here's our initial or pream RNA, or sorry, this is the DNA, but here is the primary transcript of RNA made from our DNA. So this is the transcription step. And we're copying RNA from DNA. But the colored regions here, those are the exons. Those are the coding regions. They code, they have a template for the different amino acids that are going to be added to a chain to make a protein when we do translation. Since this is one gene, but you can see if the body splices out certain coding regions, we would, if this has a certain amino acid sequence, this has a certain amino acid sequence, this one, this one, this one, they all have different amino acid sequence. If we were to splice out, if you look here, we spliced out number 3, but we left all the other exons on. And these all contain different codes. In this other protein here, we took out number 2, we spliced out number 2 and number 4. And you can see this gives us a different protein than we see here. So this is how we can get multiple proteins we do what's called alternate slicing. Okay, alternative slicing. Or sorry, splicing. Not slicing, splicing. We're splicing out the exons. Okay, so we do alternative splicing, and it can allow us to get different proteins from the same initial messenger RNA transcript. Along with making different amino acid sequences, proteins can also be modified by adding different groups. Phosphorylation, that's adding a phosphate group, or also cutting into smaller units, or methylation is adding a methyl group. You don't need to know what a methyl group is, but you can see there are adding modifications to these proteins. Here's that phosphorylation. This is the protein, the blue ribbon's the protein, but we're adding various things to them, binding various molecules to them to make different protein. Okay, even though this may be the same amino acid sequence, this one has a phosphate group, but this one has a phospholipid added to it. So this would change the characteristics. From this to this would have a different characteristic. So once we have the mature RNA form, it will be taken to the ribosomes. The ribosomes are the sites in which the mature messenger RNA gets read, and we put together the string of amino acids to make the protein. Okay? And these messenger RNA contains the codes for which amino acids are going to go and in which sequence. So like these nucleotides of the messenger RNA would be for the first amino acid here. The second, third, before the second one. Okay, so it's going to be every 3, which are called bases, which is called a codon. Each 3 bases makes up a code for a certain amino acid, and that's how the chains are going to be made. These ribosomes, okay, they're made up of a large subunit and a small subunit, and made up of ribosomal RNA and also protein. And they serve as the enzyme sites to put those peptide bonds together between the amino acids. There are 2 places you would find the ribosomes. Okay, you can find them free, floating in the cytoplasm, or associated with the granular endoplasmic reticulum, which we're going to talk about in a second. And this is basically depending on where the proteins are going to be used. Okay, the ones in the cytoplasm make proteins that are going to be used in the cell. Okay, in the cytoplasm. The granular makes proteins that are going to be packaged into lysosomes, packaged into proteins that are going to go to the membrane, the plasma membrane, to be embedded, or packaged in vesicles to be eventually secreted out of the cell. Okay, so that depends on where the ribosomes sit, what kind of proteins being made, determines on which messenger RNA goes to which type of ribosomes, either the free or the granular. Okay, and the ribosomes can exist in two different places in the cell. One of the places is it can be fixed to the endoplasmic reticulum, the ER. Okay, granular, or also you probably heard it called rough ER, rough endoplasmic reticulum. Okay, it has ribosomes fixed to it. There's also a granular or smooth ER that doesn't have ribosomes fixed. So the granular is involved in making protein. Okay, modifying proteins, putting them together, because the ribosomes exist on the rough ER. You can see here the little purple dots are the ribosomes. Okay, and I didn't say it, but the rough ER is this green, okay, passageways, the little pores here in the nucleus. The messenger RNA is made here and will make its way through the pore and into one of the ribosomes to get translated. Okay. Our agranular ER, endoplasmic reticulum, looks like this, the smooth. You don't see the little purple dots, the ribosomes on here. Okay. It serves a different function. It makes enzymes for steroid productions, stores for calcium. We'll see that when we do look at skeletal muscle and muscle contraction. Okay. So the granular ER is going to be involved in protein. Agranular is not involved in the protein synthesis. Okay. Associated with the endoplasmic reticulum here, the green, is the purple. This is the Golgi complex. Okay. The Golgi complex is basically a modification. It's kind of a FedEx. It will receive the protein that gets synthesized here at the ER. It will modify the protein and it will package the protein to be sent out. Because the proteins that are made at the ribosomes in the endoplasmic reticulum, they are made to be sent out. They don't stay in the cell. They're either going to be sent to go to the membrane, or they're going to be sent for secretion. If you remember, hormones get secreted, so hormones would be, if they're a protein hormone, peptide hormone, they would be packaged and would be packaged in the vesicles at the Golgi complex. So the Golgi complex kind of does modification of the protein and then packages it and sends it on to where the protein needs to be sent. It also can package it in lysosomes, in vesicles. So if we can get packaging in vesicles for secretion, for embedding into the membrane here, then the proteins are made at the endoplasmic reticulum and sent out. All right, so let's look at the step involved in protein synthesis. Protein synthesis, also called translation. First, that messenger RNA leaves the nucleus, either goes to the free ribosomes or goes to the ribosomes in what we saw in the rough endoplasmic reticulum. And each group of 3 bases of DNA, right, contains a complementary code of 3 bases of mRNA. That 3 bases sequence in the mRNA is called a codon. And each of those codons will contain a code for a specific amino acids. Remember, there are 20 amino acids that we have in our body that we use to make the various proteins. Okay, so again, the codon codes for a specific amino acid. Okay, so whatever the order of codons, what codons are there and what order they on determines the order of the amino acids and which amino acids get added. Okay, so that's essentially how it gets read. It gets read in order, and each 3 is a specific amino acid, so those amino acids get the specific amino acid get added in the right order. Okay, there is one on this slide here. As we look at the protein synthesis, this should be actually template strand. Okay, because the coding strand should be the same as what's over here, except for there's U in here instead of T. Okay, but you can see each of these 3 bases, all proteins start with the amino acid methionine. These are the amino acids here: glycine, serine, isoleucine. I'm not going to make you memorize all these amino acids. That's for biochem class, but realize that these have different R groups, right? And that gives the characteristic of the amino acid and also allows for the proper folding of the protein. Okay, so we're going to use this code to put our specific amino acids in the order, making those peptide bonds, right, between our different amino acids. And you can see this, it actually gets more complicated, but you can see there are AA wouldn't code for, it codes for, this is the DNA, so the corresponding or complementary RNA is 3U's. 3U's would code for phenylalanine. Okay, 3U's would not code for serine or any of these amino acids. It would code specifically for phenylalanine. Okay, so you can see the different, oops, let me go here. It's the RNA triplets, the codons here, code for different amino acids down here. So depending on what code is in that messenger RNA determines on which amino acid, and where that codon is in the code determines the order of the amino acid. Okay. You see, this will get a little more involved in just a second here. Now, this is not something I want you guys to memorize, okay, but I need you to recognize what it is for. This is the triplets, the codons. This would be the first letter. Okay, second letter is up here, and third letter is down here. Okay, and so if it's U, U, and a U, right, we already saw that at phenylalanine. Okay, but if you have U, U, and now we go down to the third and we have A, we have leucine. Okay, if you were to have this on the exam, I would put this up there so you guys have it to use. You need to know how to utilize it and what it means. You don't need to memorize this. We got bigger and better things to go after here in this class. But if you are ever to take a biochemistry class, be forewarned that you're likely going to have to memorize more of this. Okay, some other ones I want you to recognize is AUG. This is methionine. Okay, this is the first amino. All amino acids start with AUG. Okay, AUG. So they all start with methionine. Then there are stop. This tells us where the stop is. Okay, so the amino acid will start, or sorry, the messenger RNA will get started read at AUG, and then all the triplets in between the various amino acids will be added until the ribosome reaches UAA, UAG, or UGA, and that's your stop. That will stop the translation process, and that will stop the producing of that polypeptide chain for production of that amino acid. Okay, once you reach there, messenger RNA, the protein's released and can go out and serve its function. And depending on which ribosomes it's at, remember free ribosomes, the proteins are used in the cell, where the fixed ribosomes and the endoplasmic reticulum get packaged up and sent out to where they need to go through the Golgi apparatus. So the messenger RNA contains the code, the amino acids that actually bring, sorry, not the amino acids, the RNA that actually brings the amino acids to the ribosome are transfer RNA, or you'll see it's small tRNA. Okay, these are these RNA molecules. They have these hair. This is what it actually looks like, but there is a region that has an anticodon. This region is going to complementary base with the codon in the messenger RNA. Okay, so this will be the complementary nucleotides, nucleic acids, bases to the actual codon in the messenger RNA. Okay, and then it has an amino acid accepting in. This is where the amino acid will be bound to. Okay, depending on what codon, anticodon here, depends on what amino acid is here. Because remember, the codon codes for a specific amino acid. And so the anticodon, the complementary, would only hold on to that specific amino acid. Okay. So just looking at this anticodon, UAC, okay, this is transfer RNA. It will complement with the complementary messenger RNA. So U would complement, would be a messenger RNA would have A would have U, and C would have G. So that would be the start. Okay, so the amino acid that would be attached here would be methionine. Okay, so UAC, okay, there it is. The methionine would be the amino acid that would be attached here, not phenylalanine, not cysteine. It would be a specific amino acid because this anticodon lines up with this codon in the messenger RNA. So this just shows, written showing the different steps involved that we just talked about. First, the messenger RNA goes to the ribosome, and the tRNA attaches to the codon because the tRNA has that anticodon. Right? It will start first at the start codon. The amino acids are attached to the tRNAs, form the polypeptide bonds. Okay, the tRNAs will disassociate from the mRNA as they lose their amino acids in that chain that's being catalyzed at the ribosomes, making the polypeptide or the protein. Okay, and this will all continue in the ribosomes, reading the mRNA, bringing the tRNA in until we reach that stop codon. Right, and then the whole complex, the messenger RNA disassociates and the protein will disassociate. Okay, if you remember, our proteins, they have to fold. So as they get released, the amino acids cause bends and folds, and making that secondary and tertiary. And this is just a visual showing the messenger RNA, okay, coming through to the ribosomes, and the tRNA bringing in the amino acids. So codon 1 would associate with this amino acid, right? And it would be a specific tRNA that would bind there with amino acid 1 that was in place. This codon here would associate with a different tRNA that would be holding the number 2 protein. So this is the process. The mRNA gets read, tRNA comes in, and the amino acids get bound to each other as they are brought in, and we get this growing chain. Once we reach the stop codon, the protein chain will get released, and the mRNA will get released, and then as we're making this, the protein gets folded. At the end, it will get fully folded, and now we have that fully functional, once we have it folded up, we have that fully functional protein. I'll try to post some more also. I know I've probably made the caveat in Zoom, but the internet, you guys have lots of internet resources. I'm going to try to pick up some various ones for you guys, but you have the world. When I took this class, basically we had the book and our lectures, and that's it. But you guys, especially in physiology, because it's a very dynamic, it's not like anatomy where it's very stagnant. There's stuff moving around, molecules moving around. So watching it in a video actually, will help you dramatically. So definitely use those resources. And again, I will try to post them too, but sometimes you might need more. You guys got the internet, you can find them at. So please utilize those. Now we made a protein, let's look at another copying of DNA. We've copied DNA to messenger RNA, but also your cells throughout your body, throughout the lifetime, there are many cells that have to replenish themselves. Your skin cells, they are not the cells you were born with. You have cells, stem cells underneath that are dividing to help replenish the cells that slough off your skin. Okay, so there are many cells in your body that have to be replenished. So we can look at replication that we'll be talking about in DNA. We talk cell cycle. Okay, I'm not going to go over all these stages, but you can see the cell cycle. Okay, you've probably seen this in anatomy. We have a G1 phase, an S phase, a G2 phase, and then mitosis. Okay, and cytokinesis. Mitosis and cytokinesis are part of the mitotic phase, but they are two separate events. Okay, they will be taking place at the similar times, but two separate events. But let's look at cell cycle, and this can vary for your different cells of your bodies. Okay, your neurons. When you get a spinal cord injury, do you replenish? Do you usually get use of those cells that were killed off? No, they don't get replenished. So the cell cycle for a neuron is basically your lifetime. They survive throughout the life. They're not replenishing, although they have found small pools of stem cells. For the most part, most of your neurons are not going to divide and replenish themselves. Okay, so those cells, they stay in. Okay, this whole process is interphase. Okay, the neurons will stay in G1. Okay, this is just your normal everyday activities. Okay, but those cells, the cells that need to divide, the most rapidly dividing cells are actually those that line your gut. Your gut has lots of friction going on, lots of food you bring in, so they get damaged. They have to be replenished. So those cells divide fairly rapidly. The stem cells divide to replenish those cells that are getting sloughed off or getting damaged. Okay, so those cells that aren't there for a lifetime will start going into these phases. Okay, first we're in interphase still, but the S phase is involved in DNA replication. Okay, because all your cells have 23 pairs of chromosomes. They must have 23 pairs of chromosomes to be viable, okay, or to be considered normally viable. Okay, to do that, if we're going to take one cell and divide it into two, we can't just divide the chromosomes up, because then each cell would have half the amount of chromosomes. Okay, so we have to first do DNA replication. We need to make a copy of the DNA if we're going to divide the cell. Okay, because your stem cell is one cell and it divides and produces two cells. One cell is still a stem cell, the other one becomes the cells that are going to help replenish whatever cells are sloughing off or dying off or that need for growth. Okay, so DNA replication must take place because we need to make an exact copy. All your cells have the same DNA, so we make an exact copy of the DNA. Okay, that's still an interphase, but S phase. These are the cells that are heading towards dividing. Okay, in the G2, we get more growth, and the activity before mitosis will make proteins that are needed because we're going to be separating the cytoplasm and the DNA, okay, to divide the cell. Okay, so here's our interphase. Once we get past there, now we hit mitosis stage. That are these here. You've likely went over these in anatomy. The prophase, okay, metaphase, anaphase, and telophase. Okay. These are separate events, but mitosis, the mitosis event is involved in dividing of the DNA, dividing of the chromosomes. Okay. Happening alongside at the end of mitosis, but happening the same time around telophase of mitosis, is cytokinesis. Cytokinesis is dividing of the cytoplasm. Okay, happening similar times, but two separate events. Mitosis involves dividing the DNA. Cytokinesis involves dividing of the cytoplasm. Okay. So as we already showed, interphase involves normal activity, also preservation for those cells that are dividing. And then the mitotic phase, don't confuse that with mitosis, would include both mitosis and cytokinesis. But mitosis is an individual event compared to cytokinesis. Mitosis involved in division of DNA, cytokinesis, division of the cytoplasm. Since you've already had most of the stages and stuff in anatomy, I'm not going to repeat, but I'm going to go over the process of replication. Because dividing, we have to replicate the DNA. Okay? For transcription, we're taking DNA to messenger RNA. Basically, with replication, all we're doing is making an exact copy. So we're making a two-stranded copy of DNA. Okay, and it takes place before cell division can take place because we have to have 23 pairs of chromosome in each cells. Okay. In transcription, you saw we had RNA polymerase. Okay, we're going to have a DNA polymerase, but we have a second set of enzymes, and those are the helicases. Okay. In transcription, the RNA polymerase both broke down the hydrogen bonds and also formed the bonds for the RNA, the messenger RNA. Here we have a helicase enzymes, and these are involved in breaking down the hydrogen bonds between the DNA strands. Okay. This creates a fork because we have to have access to those bases to be able to add the complementary bases to make a new strand of DNA. All right, so after the helicases breaks up the bonds, all right, it makes that bubble where we can add the new nucleotides to make a new strand. Okay. The two new strands will be made up of half old and half new DNA. Okay. And this is called semi-conservative replication. So let's say this is the old 1, old 2 chromosomes. Because remember, they're made in pairs. This is the new. All right? The replicated DNA will not look this way. It will not have 2 new strands for one and 2 old strands for the other. It will be like this, this. Okay? It'll be half old and half new. Okay? Although this new strand is an exact copy of this old strand, and this new strand is an exact copy of this old strand. So this DNA is identical. Oops, I won't write it here, it'll take too long. Identical, but at least base pair wise it's an identical, but it will have half old and half new DNA strands. We're using one strand to make a new strand, and we're using the old strand here to make a new strand. So we're going to have, once we're done, this will all be the 2-stranded DNA consisting of this old and this new. This DNA will consist of this old and this new. Okay? But this new strand made here will be exact copy of the old strand here, and this new strand being built here will be the exact copy of the old strand there. Okay, you can see the DNA polymerase. The helicases unzip this to give us this bubble, and then the DNA polymerase works in, okay, can only work in one direction on the DNA. It works this way to build, and then this way to build. Okay, but again, we get semi-conservative. We get one strand old, one strand new. And here's showing what I just kind of drew earlier. You see the old strand's the blue, the red strand's the new in this case, and you can see the new replicated strand is the same as the old strand, and this new strand is the same as this old strand, at least pairwise, okay, at the base pairwise, and that gives us semi-conservative.